Yes.
Hi, my name is Leho Tedersoo, and together today we are collecting a sample for FunAqua project, which means core sampling of water and sediment below. So for this we need a syringe, some ethanol, a bottle of clean water or an empty bottle. We need gloves, some beakers, and also a water filter. Some of these can be done also back in the lab. So get into the water, we need to put on the gloves because we need to collect both the water and the sediment, and we need to keep it as clean as possible. So we can rely on the bottled water, and we collect this lake water here. And for the other tube, I will collect the sediment. So we collect our water sample from about 40 centimeter deep from the coast, and we collect the water from the depth of around 30 centimeters, so about 10 centimeters above the surface. Second, we will collect the sediment sample. So this is a 50 milliliter tube, and we will fill half of this with a sediment. For collecting a sediment, we scrape off the surface of the bottom, and from about 5 to 10 centimeters depth in the sediment, we pick this single sample. So it's good to take with the gloves on, and this way we can also get rid of the excess water, and we fill about half of this tube with the sediment. Okay, so it's half filled. Now we will add ethanol on top of it, so it will kill the bugs and also preserve the sample for DNA analysis. Ethanol can be also substituted by some more specific preservative. So this is the sediment sample now, and we will take it back to the lab. Now I can take off my gloves. So that was the water sample. Now I'm taking the syringe and the filter. So the filter will be screwed to the syringe, and we are going to pass through 150 milliliter of water. So it means 3 syringefuls. So 50 right now. So all we need to do is to lead the water through the filter Then to get it back, we have to unscrew it for a moment. Then we can take off the syringe just to evade the vacuum that is generated inside the syringe. And we go with the second flow. Second 50. Again we force the water through it. So if the water is very murky, you can also deal with less water going through the syringe, but you will certainly have to make a note about the amount of water going through. And now it will be the last syringe full. So, as this was the last, and there is always some remaining water in around the filter, so we can get rid of it just by rattling it strongly, and we can also play with the syringe so that we push it and rattle it at the same time. Then we can get rid of the last. Now we will have to add some ethanol also here, or another preservative to maintain this sample for DNA analysis. So first we need to screw a cap to the filter end. Now I take the filter off to access again the syringe. I will put it on, and now the important step is to fill it fully with ethanol. So we add only a few milliliters of ethanol here, just to make this filter compartment full of ethanol. So we can make it sure just by gently, very gently pressing it. Now the filter tube is filled with ethanol. We can get rid of the rest or drink it. And now we will also fill the other side with a similar cap, but we will screw on the other side of the cap. It has two sides that are both filling it. So now it's the sample. And finally we have a leftover water, so we will either collect it into the same water bottle, keep it here, or take it into an empty flask. This 50 milliliter is enough for any type of water chemistry analysis. So it's better to keep the water at room temperature, because it will break if you put it to -20. But those sediment and filter samples, these are best to put at -20 for storage before shipping to us in Estonia. So, thank you for attention.